Alright, hey everybody, welcome back to another week of zoology. And so today we'll be going over two different phyla, the phylum Annelida and then the phylum Mollusca. So we'll be going over three classes in phylum Annelida first. We'll be going over the oligochaetes, so terrestrial earthworms. We'll be going over polychaetes, so marine worms, and then we'll also be going over the class HIRUDINEA, such as leeches. Alright, so first, before we get into the first phylum Annelida, I'll go over a little bit of what brings us to this point in our phylogenetic tree. So we've gone through the phylum Cnidaria, we've gone through the phylum Porifera, briefly touched on the plantiflagellates, and then last week we went over the phylum Platyhelminthes. And so now we're up to these two right here, the annelids and the mollusks. So both these groups are protozoans, so the first opening in that developing bundle of cells is the mouth, and then they're also considered lophotrochozoans. So at some point in their development, generally very early on in their development, during their larval stages, they'll have this ciliated band which draws food closer to its mouth. So both of these groups, the phylum Annelida and the phylum Mollusca, fall under these two categories of being a protozoan and a lophotrochozoan. We now have segmentation for the first time. So annelid means segmented worm, roughly. And so you'll see that each one of its, well, it has a various amount of segments, and each segment has a various or special job that it functions as. So I just mentioned that they're lophotrochozoan protostomes. So once again, it has that ciliated band around its mouth while it's in its larval form. It's free floating. And then it's also a protostome, so that mouth is the first opening to the digestive system while it's still developing as a bundle of cells. I mentioned segmentation, and so each one of these structures here, so at the very top of the screen, you can see that structure here. Let's see if I can cut it off. So that is a single segment. And so if you look at an earthworm, it has multiple links of these segments, and each one of these segments is separated by a layer called a septa. It's this really simple cuticle, very thin, that separates each one of these cavities inside. I went over paired metanephridia last week, but I'll go over it again. There are these two openings within each of these openings in the body cavity. So in each segment, there's an opening or two openings that lead to these metanephridia. Those openings then lead to a series of tubes that run past capillary beds. And so as body fluids flowing past those capillary beds, any excess water or nutrients that might be wasted gets reabsorbed back into the body. Waste products continue out through these series of tubes and then leave through this structure right here called the nephridia pore, and there's one right here too. So they have a fairly complex excretory system, much more complex than the protonephridia that you saw 2 weeks ago in the planaria, or last week when I discussed it a little bit. And so for the first time, we have closed circulatory systems. So we have true blood vessels that are pumping circulatory fluid throughout its body. It can now send nutrients or oxygen to different parts of its body using the circulatory system. And so you'll see in our first dissections of this week, the dorsal vessel, which is carrying fluid down its body. We also see hearts, which are actively pumping this fluid throughout its body. Another thing that is unique is we have a cuticle, which is a thick, waxy outer layer. So if you look at this diagram on the side, it looks like the skin of this earthworm. That's actually the cuticle, and like I said, that's a waxy outer layer, and so that's used to prevent this earthworm from drying up. It keeps everything inside of it. That's a terrible analogy. It acts as a barrier to desiccation, so drying, and stuff like that. We now have a coelom. So a coelom is a true body cavity. So we have our body, our digestive tract, and then we have body cavities within or outside of that. So each one of these segments is separated by a septa, and then within each of those segments, there's this body cavity, and then you have that digestive tract that's running through the length of that body. And then within this digestive tract, you have what's called a typhlosoul. See if I can typhlosoul. I might have it written on another slide, but you guys will see it Wednesday or Thursday when you guys come in for lab. But this structure that hangs down, it looks kind of like a tonsil right there. In this diagram, so that's right here, that's the typhlosoul, and so that's acting as a way to increase the surface area of this digestive tract. The more surface area you have, the more space you can, your enzymes can work, and you can digest more food. So they're monoecious. They produce both male and female gametes, so hermaphroditic, and then they use this clitellum, which is covered up by my little video, but you'll see it in person on Wednesday or Thursday. And so they use this clitellum as a way to direct mucus, and they use that mucus to kind of roll through past their cuticle, collecting any excess sperm or eggs, and then directing it to their oviducts, so where they then fertilize. So we're going to jump right into our classes. So in the phylum Annelida, there's 3 classes we'll go over. There's the phylum Polychaetes or POLYCHAETA. There's the phylum OLIGOCHAETA, and then there's the I'm sorry, not phylums, class OLIGOCHAETA, class POLYCHAETA, and the class HIRUDINEA. So our first class, the class POLYCHAETA, is the most ancestral group in this this phylum. So our next class that we'll go over, the class OLIGOCHAETA, evolved from these polychaetes, and then went into freshwater systems. And then from there, our last class, the class HIRUDINEA, are the most derived, and so those evolved from the oligochaetes. So there's roughly 10,000 some described species in the class POLYCHAETA, and so it's the most diverse class in this phylum. Only about 170 are found in freshwater habitats, so just under 2% are freshwater. Everything else is completely marine. And so one way you can distinguish the class POLYCHAETA from the class OLIGOCHAETA is they have these parapodia, and so these structures right here on either side of the body. So one right there, on this side, and so those act as fans almost, or like small appendages or arms, and so they can control these, and then they can pull themselves through the water, kind of like small legs on a millipede or something like that. This also helps with respiration. So these appendages, these parapodia, are increasing surface area of the body of this organism, and so they still rely on diffusion for oxygen to come into their body and CO2 to leave their body. So when you have this increase in surface area, you can live in the soil and the sediment of a marine environment, so in the benthic layer of a lake or of a sea, you can crawl through with these parapodia, and then you're also absorbing any excess or oxygen in that environment that you can. So it has a dual purpose. It serves that purpose fairly well. They have a fairly distinct head. I don't see any on this slide. So most of the times the adults will have fairly well-formed jaws. So if you've ever heard of a bobbit worm is an ambush predator. So it'll sit inside the soil with its head just sticking outside the water, and then it'll have its jaws just above the sediment like that. And then when some poor fish or whatever floats by, it'll reach out and grab those that prey item with those jaws. So I included this one kind of last minute because I think it's the most insane name I've ever heard of. So here we have Osedex mucofloris. So it's found in these whale falls. So a whale fall is when a whale dies. It doesn't just wash up on a beach at all. It'll just sink down to the bottom of the ocean, and so that's an enormous amount of organic material, fresh organic material that's being placed into this relatively nutrient scarce environment. So you have this just feast of organic material, and one of the first colonizers are these polychaetes, Osedex mucofloris, and so these are found specifically in the bones, and as such they're named the craziest name I've ever heard. They're the bone-eating snot flowers. So if you look at their name, Osedex, Ossie. So if you have Osteichthyes, or the bony fishes, Ossie means bone. Muco obviously is snot, so you have mucus, and then floris is flower. So whoever named these was very creative, but was not very endearing. So the bone-eating snot flower is quite the term. But yeah, I had to include that when I read about this organism. So they're fairly neat. And one subject of research that these organisms are being really questioned about is how they find these whale falls. So they've dropped dead cow carcasses in the ocean to try and control how these polychaetes find them, but they still are not sure. They think they just kind of float around as that Lophotrochozoan in that larval form, and then they land upon these whale falls just by sheer dumb luck. So it's a neat organism with a terrible name. The next class is the class of OLIGOCHAETA. So these are the earthworms. These are the terrestrial segmented worms. So if you want to compare these directly to the polychaetes that you saw in the last couple slides, these don't have as much setae. So on those polychaetes, they had very dense rows of setae along those parapodia. You'll see in our dissections Wednesday or Thursday that these OLIGOCHAETs have very few setae. If you run your finger against them, you will feel those bristles, but it's definitely not as much as you'll see in the specimens that we have in jars. They don't have parapodia, so if you look at each one of these earthworms, they don't have any appendages, any fleshy appendages that are going out to the side, but they still have cutaneous respiration. And one of the ways that they're able to do this is that they have they live in really moist environments. So you have to have some layer of water or mucus along the outside of your body for for oxygen or CO2 to diffuse into and out of your body. So they live in these wet environments. So moss in the soil, stuff like that, in the benthic layer in freshwater streams or lakes. So they don't have a really distinct head, and so you'll see on Wednesday or Thursday, the OLIGOCHAETs that we'll be looking at have what's called a peristonium. So it's this burrowing adaptation that covers their mouth, but it's very difficult to tell one end from the other, besides which side has the clitellum, and so the clitellum is this structure right here. It's this really fatty layer right in the center of that earthworm. And so they use this to, I may have mentioned this earlier, they use this to create a ring of mucus that they then collect all male and female gametes, so both sperm and egg, and they push it towards their oviduct on the underside of their body. So once again, they're monoecious, so they produce both male and female gametes, but they also reproduce with other individuals. So they kind of wrestle each other and then swap gametes. And they use this cuticle to produce the mucus and then push that mucus towards those oviducts. So this is the class HIRUDINEA. These are the leeches. There's roughly 680 described species, and most of which are found entirely in freshwater habitats. In fact, I believe 90%, 99% of them are found in freshwater habitats. And so of those roughly 700 species, almost 500 of them, or 520 some of them, are parasitic, and the remaining 200 are predatory. So they're not really found in the environment or the freshwater around here, but if you go north to like Michigan or New York or Maine, those lakes are just filled with them, and those are the parasitic ones that you'll find. You'll also find them in tropical areas, and so this is a neat way to track mammals. So it sounds weird, but you can track rare species of mammals using these leeches. So if you have really dense underbrush or really difficult terrain where you can't find your target species, whether it's a rabbit or whatever, you can take these leeches from any aquatic environment, and then you can check their gut content for whatever blood they've been feeding on. And so the mitochondrial DNA that's in their gut content can still be replicated up to 4 months after they dropped off of their host. And so from there you can determine which species of mammals were in that area. So you don't have to spend so much money trying to find these mammals as much as you just have to find these leeches. So it's a pretty neat use for parasitic leeches, scientific use at least. So if you want to differentiate these leeches from the other two classes, they don't have parapodia. So if you look at a polychaete and a leech, all you have to do is find the parapodia, and you know you're in the class POLYCHAETA, whereas the other organism is in the class HIRUDINEA. So they have a relatively flat body, so it kind of looks like an arrowhead, flat on one side, and then it kind of tapers to a point. And so they have suckers at both sides. So there's a sucker on one end here, and there's a sucker on the other end here. And so they use those suckers to pull themselves forward while they're moving. So they kind of reach out with their head, which is the smaller side here, and then they reach out with that head, they grasp onto a point with that sucker, and then they contract their body. They release that hind sucker, and then they contract that body to the head, and they just pull themselves along like that. And they also use that head to attach to hosts such as humans or other mammals or reptiles, anything that's warm-blooded living in those freshwater, or comes across in those freshwater environments. So once again, most of these are parasitic, 500 species. You've probably seen really old video or movies about tropical expeditions where they just enter a pond and then they walk out and they're just covered in leeches. And so that's somewhat true. You can pull them off by hand, so it's a myth that you have to use salt. You can use salt and they will fall off, but you can use your hand because they don't have true teeth. So they just are using that sucker and they don't actually burrow into your body. So you can pull them off by hand if need be. That brings me to the second phylum for today. So we have the phylum Mollusca, and then we'll go through 4 classes: the POLYPLACOPHORA, the class GASTROPODA, class BIVALVIA, and class CEPHALOPODA. So once again, we're looking at Lophotrochosome protostome. So they have that ciliated band in their larval stage, and so they're using that cilia to push food towards its mouth. And then they're a protostome. So while they're developing that bundle of cells, the gastrula, the first opening to the digestive system is the mouth. So this is an extremely diverse phylum. There's 90,000 species. Everything we've covered before this point has been roughly 20,000 species or 10,000 species. So this is an extreme jump, and most of these are actually gastropods or bivalves. So slugs, snails, or mussels. And so the difference between the phylum Mollusca and the phylum Annelida is that we have some sort of shell being secreted by these organisms. And so this shell is used for either protection or structure. And so most of the times it's made out of calcium carbonate, same structure that you saw making the spicules in the class Calceospongiae in the phylum Porifera. So we still have that paired metanephridia that I discussed 8 slides ago for the phylum Annelida, and so we still have that same nervous system or excretory system. We also have both monoecious and dioecious species, so you'll have some species that are producing both male and female gametes, or you'll have other species that are just producing a single gamete, whether it's male or female, depends on that individual. We have a complete digestive system, so we have an opening for the mouth and the anus, so we have 2 distinct openings, and the same thing is found with the phylum Annelida that you saw a couple minutes ago. They have both a mouth and an anus as well. So another difference between the phylum Annelida and the phylum Mollusca is we have an open circulatory system. So instead of having all the fluid pumping through vessels, such as a vein or an artery, we have an opening, and then blood or hemolymph flows around an open cavity before it re-enters either the heart through a point or through another artery. And so you'll see that on Wednesday or Thursday when we begin our clam dissection. You'll be able to see the heart and the pumping vessels, and then you'll notice that it's just pumping fluid throughout its shell without any veins. So it doesn't have a true circulatory system, but it's open. Alright, so the first class that we'll go over is class POLYPLACOPHORA, and so these are the chitins. And so instead of having a single shell like you see in most snails, or 2 shells like you see in bivalves, they have these rows of shells. So you can see on the back of this chitin, it's got 1, 2, 3, 4, 5, 6, 7, 8, 9, 9 shells along its back. And so each of these are individual plates that are being excreted by that mantle. So they're made of calcium carbonate, and they aid in the protection of these chitins. And so these chitins live in relatively shallow marine areas. So they just kind of go along the ocean floor, along rocks, in tidal zones and stuff, and they just start scraping up algae and then eating from there. There is a really cool specimen. It's my favorite specimen in this entire class. This is Cryptosis stellari. This is also the gumboot chitin, or the giant fiery wigwam chitin. It's a really neat specimen, and so this just shows how large a chitin can get. This is roughly, I don't know, a foot long. So I mentioned it's my favorite specimen. There's a lot of cool stories about these organisms. So the one that I like to repeat is there was a ship that was stranded or shipwrecked. I believe it was like the Aleutian Islands, so off of the coast of Alaska, in that island chain. And so the sailors were starving, looking for a food source, and they came across these gumboot chitons. And so they tried to cook them up, and it turns out these organisms are so unpalatable or disgusting tasting that they decided to starve and wait out rescue while not eating, as opposed to eating these chitons. So, like I said, this is my favorite specimen just because of how unique it is and the size of it as well. So, the next class is the class Gastropoda. So these are the snails and slugs. I believe everyone here has seen a snail or a slug at some point in their time. They're very common, and so this is the largest and most diverse class in the phylum Mollusca. You have all three major environments represented. So you have saltwater, freshwater, and terrestrial environments represented. So here, these three, so 1, 2, 3, those three are saltwater snails. If you were here last semester, we had a tank, and we had several conch snails, or not conch snails. Yeah, maybe they were. But anyway, we had some saltwater snails, or some marine snails, and so they would eat these. They are predatory, and so they do have a little harpoon, and so they can sting with that. So they were pretty neat to watch and feed at the same time. But you all are probably much more familiar with the freshwater or the terrestrial snails and slugs. So one of the most common ones where I'm from in Maryland is called a periwinkle. I don't know if you guys have them around here. I haven't seen any. But those are these small snails. They look kind of like light gray with a really spiraled shell, and so they sit on these grasses at low tide, and as the tide rises, they climb up the grass to stay out of the water, and then as the tide falls, these periwinkles move down the grass, down the stem of that sedge, and start eating all the algae and the diatoms and whatever's left on that stem. So there's a lot of neat organisms. Once again, it's the largest and most diverse class. So the difference between snails and slugs: snails have a shell, slugs do not. So snails have slugs have these the mantle. So the mantle is what secretes this extremely hard shell. So they have this mantle, but they don't secrete that shell. The next class is the class Cephalopoda. So just like snails, not just like slugs, they have a mantle, but they don't secrete an extremely hard shell. So the organisms in this class include squid, octopi, cuttlefish, and a nautilus. And so these are extremely intelligent organisms. It's kind of a cruel thing to put them in an aquarium and not give them any sort of stimulation. So oftentimes when you see cuttlefish or octopi in aquaria, they're either extremely depressed, looking for some sort of mental stimulation, or if you go to a good aquarium, they're well taken care of, and they don't have that issue. But if you have your own personal aquarium and you try and keep octopi, they do end up getting depressed because they don't have anything to do. So you have to keep giving them puzzles and such. There are a couple neat stories about octopi that are escaping their enclosures because they've memorized the schedules of their caretakers. And so once again, these invertebrates are extremely smart. One of the things that aids in these organisms' intelligence is that they have a very complex eye. So this is one of the 3 times that an eye has evolved over time. So it's evolved in cephalopods, in insects, and then, or I'm sorry, in cephalopods and arthropods, and then in vertebrates. So here, the major difference between a vertebrate eye, so an eye that we have, and an eye that a cephalopod has, is that they can actually change the size of their eye while you're just changing the amount of light that enters our cornea. They're actually focusing their eye, changing the size of it in order to focus on an object. So it's very neat organisms. Once again, my bad. They're male. They have separate males and females, and so we have one of the specimens in here. We have a female octopi or octopus with one of her children. One of the neat aspects, another neat aspect about these cephalopods, especially the female cephalopods, is that they can go for months without eating while they're protecting their eggs. So these larger octopi, octopus, I always forget which one's singular and plural. Octopi is plural, octopus is singular. So anyway, with these larger octopus, they'll form a burrow under a rock, and then they'll attach these chains of their eggs to the top of that rock, and then they just won't move for months at a time. They won't eat, they won't react to anything except to protect those eggs. And so once those eggs hatch, they die. So it's kind of a shame to waste such an intelligent creature, but that's the life cycle that they've been selected for. So, neat organisms. There are tons of cool videos online. If you just search up YouTube and you look up cephalopods changing color, or cephalopods, I don't know, camouflaging. And so one of the neat videos I used to show was of a octopus that had been scared. There was this diver that comes across a piece of coral, or an object that looks like coral, while out on a dive. And so as he comes up to it, almost the entire thing shrivels up and turns into this white octopus that just swims away as quickly as possible. So they have these incredible ability to change the shape of their body, as well as change the color of their skin, and so they can do that using what's called chromatophores. And so one of the things that are cool about these cephalopods is that they change colors based off motion or based off camouflage. So if you ever look up, there's a really neat video of a cephalopod or a cuttlefish in an aquarium. And so the guy that's filming it shows a picture of another cuttlefish, just like a Google image of a cuttlefish, to these cuttlefish in the aquarium. And so one cuttlefish recognizes that this picture of a cuttlefish is not one of its own, and so it comes up to the glass and gives it like a defense or a warning coloration. And then you can see all the other cuttlefish in the background noticing this one individual, and then coming up to investigate as well. So they're neat organisms, and something that if you're interested in marine life, you'll probably be extremely interested in these cephalopods. So the next class is the bivalves, and so these are the mussels, the clams, the oysters, and scallops. So this is an extremely diverse class. It's found in almost all freshwater and marine environments. The mantle extrudes these two shells. You have a dorsal and a ventral half. So if you just take this line right here, this top half that's the dorsal side, and this here is the ventral side. So dorsal meaning top, ventral meaning bottom. That shell is made of calcium carbonate once again, and so it's fairly rigid. And then they can use these abductor muscles at the back of those those shells where they meet, and there's a hinge, and so they can open them rather slowly, but they can clam them closed with a lot of force, and they can keep them closed using those adductor muscles. So if you're a raccoon and you're trying to really crack open this this muscle, you're going to have to spend a lot of time and a lot of effort to do so because of those abductor muscles at the back of that shell right near the hinge. So it's a little bit of physics. So if you imagine you're swinging a door open, if you swing near the door handle where a door handle normally is, it opens with a lot of force. But if you try and swing it open near the hinges of the door, it takes a lot of force to just push it open very slowly. But if you try and keep it closed, it's much easier to hold it closed by the hinges than it is by the door handle. So it's kind of a neat biological adaptation representing physics, real life physics issue there. So these mussels, these clams are they're sessile, so they don't move. They can move. They have a foot that they can exude, and so, but they're not moving at such fast rates that they can readily move to new habitats if they're exposed to some sort of pollution or some sort of issue like a stream drying or something. So in order to overcome these difficulties in movement, they take advantage of that low photrochozoan larvae. So that free moving, that bundle of cells with the cilia around the mouth, that's free floating in the water column. And so they take advantage of that. And so they have special adaptations to get their larvae up into the water column and then move that water or that larvae upstream. So if you think about it, while you're in a stream, it's very difficult for organisms that are moving only a few inches every year upstream or downstream to get their offspring upstream. So without any of these adaptations, there would be no mussel life or bivalve life in any stream. So water currents pushing it closer and closer towards the oceans. And if it doesn't have any adaptations to keep moving it farther upstream, then it can't take advantage of these habitats. So one of these adaptations is creating one of these lures. So this is using the foot of a mussel, and it will attract one of these fish here closer. A fish will come in and try and strike the fish lure. In doing so, this mussel will release a lot of its parasitic larvae into the gills of this fish. And so here's a neat video, let's see if I can get to play, of this happening. It doesn't have that fish lure, but this mussel uses those abductor muscles to close the clamshell really quickly, trap that fish inside that shell, and then it releases all these Glochidia larvae. The fish is like, "Why? I told you not to go near that." But everything you see here, all that white foam, that's the larvae of these mussels. And so as it's trying to struggle, it's getting more and more of the larvae stuck in its gills. So as fish breathe, they drop that buccal cavity, the very bottom of their mouth, they draw water in, and then they pump it past their gills. So in a highly stressed situation, which this fish is in, it's breathing fast, which you can see here. It just stopped. But if you rewind and play a little bit, you'll see that fish is breathing really heavily. And so as it's breathing heavily, it's drawing oxygen, or it's moving its mouth fastly. It's drawing water into its mouth and then out its gills. And so those Glochidia larvae are being drawn in its mouth and then clamping onto its gills. So a neat one of the ways that these Glaucidia larvae do this is that they have these hooks. So they look like really miniature mussels, but they have these two hooks at the very end. So right here, so this is SEM imaging of these Glaucidia larvae. And so it uses these two hooks to basically clamp onto a gill, and then it won't be shaken free by the fish. And so they're parasitic on these fish all the way until they drop off. And the end goal of this is that these mussels, these larvae, are being transferred upstream with that fish. So many of the fish migrate upstreams in order to spawn. So they face the same issues that these mussels face, with the constant flow of water flowing downstream against them. So I put a map up here just to show you all where you live. So we live, I guess Bowling Green's what, right here? Let's see if there's an X. So anyway, this is an extremely diverse area for freshwater mussels. In fact, I believe of the 250 some freshwater mussels in North America, 180, or roughly 60%, live in the mobile river basin of Alabama. And so many of the rivers that we see don't flow in this direction. They flow towards the Mississippi, but they still share many of the same characteristics, such as it being unglaciated, relatively warmer, and having a high diversity of habitat, such as log jams or what is it called? Boulders. So lots of in-stream habitat available for these organisms. So this map doesn't just represent freshwater mussels, but it also represents freshwater fishes. And so this area of the central, I don't even know what you would call this, South Central North America, is really a hotspot for diversity. There's a couple major issues that these freshwater mussels face. So they're not moving at all. So an adult's sessile its entire life, is not moving unless it releases those Glaucidia larvae out to gills of a fish, and then at which case it can then move where the fish is going. But these mussels do have the issue of being sessile in their adult's life, so they're really susceptible to anthropogenic impacts, so human-induced impacts on the environment. And so if you look at these mussels, they're really susceptible to sedimentation. So as we plow fields with our steel plows, we're disrupting that topsoil, and that topsoil flows into these streams, where it then can cover up these mussels, reducing habitat even more. And so another threat to these mussels are dams. So if you dam a section of a river, you're preventing the passage of those fish upstream to any upstream parts of that tributary. And so you're limiting the amount of habitat that these mussels can reach. Another thing that's a major problem with dams is what's called winter release. And so as these dams are built, it's obviously much higher than the water flow below them. So you'll have 50 to 60 feet deep of a lake, and then a very shallow stream below it. And so as they release water, the water that is being released is much cooler in temperature. So it's not being warmed by the sun. It's not the same temperature as is just above that reservoir. So it's much cooler. In fact, it's so much cooler that it disrupts the reproductive habits of these mussels. So these mussels don't sense that temperature change, that natural change in the seasons, so they oftentimes miss reproduction, or reproduction is late, and by then those host species of fish have either already migrated upstream, or they've been extirpated from that stream reach by the presence of that dam. So these freshwater mussels are among the most, in fact they are the most endangered organisms in North America, and probably the world as well. That video I showed on the last slide, the mussel that's in that video is called, I think it's a snuff box. Yeah, I think it's a snuff box mussel, and so that's another one. In fact, that is an endangered species of mussels. We don't have any endangered species, but we do have a couple rare species. So we have the shells, such as a pistol grip mussel, which has a really neat shell. But once again, these freshwater mussels are extremely threatened with human impacts. And so there's organizations like the Nature Conservancy, or Chesapeake Bay Watershed Association, or the Tennessee Aquarium. They're trying to restore these habitats for these species and protect diversity, especially since the highest amount of diversity is right here in the South Central America. So Alabama, a little bit of Georgia, and then the majority of Tennessee and Kentucky. So you guys live in the hot spot for freshwater diversity, and so it's necessary to protect that. So when you come to lab on Wednesday or Thursday, make sure you're wearing closed-toed shoes. If you're not, you can't come in and do the lab. And so this lab is our first dissection, 2 dissections of the year, or the semester. So we'll be dissecting an earthworm, representative of the phylum Annelida, class OLIGOCHAETA. We'll also be dissecting a mollusk, so a bivalve called a quahog. If you imagine the bar from Family Guy, the sign on top of that quahog, or I believe maybe it's the town that's named Quahog. I don't know, but either way, they're named quahogs. They're clams, and they'll be one of the dissections that we'll be working with. So after that, we have our nematode and arthropod lecture, so the ecdysozoans. That's one of my favorite lectures to give, just because I get to talk a bit about arthropods. After that, we have our midterm in, well, really it's 2 weeks from Wednesday or Thursday, whichever lab you're in. Dr. Linesh, Michael, the other TA, or some of you all it's TA, we'll figure out the best way to present the midterm to you all. The way it stands now, I'll be having review sessions. I don't know the dates yet. They'll probably be Monday or Tuesday or Friday before that midterm exam. And so I'll discuss that more in class this week, so we can schedule it in advance for you all. So those review sessions will just be time for you all to come in, look at each of the specimens, look at any of the dissections I've made, and ask any questions you have on any of the material presented up until that point. So other than that, watch the videos, take the quiz. I'll see you in class Wednesday or Thursday.